times the data has to be reanalyzed by other people for a variety of reasons. And if they can't be reanalyzed, then the value of the data has essentially dropped from a very high price, the price that it costs to generate it, to essentially zero. And then other people may have to go ahead and apply for funds to generate the same amount of data, same type of data. And that's a complete waste of time and resources. And that has to change. We can't afford that. So I know that a lot of, I don't know what it's like in Brazil, but there's a lot of people in Europe now that benefit immensely from the access to data that has been published. Much of my research now is using completely other people's data, where I reanalyze the data and extract new value out of it. And I would be surprised if a very large proportion of you guys are doing exactly the same.
Yeah, great.
So anyway, that's just an introduction to who I am. I wonder whether we should get on with the lecture.
Yeah, I'll have to wait a little bit because people at now, they are looking for where's the link, so they go to the website, get the link and come. Usually we go mostly in some 5 minutes, but we go like popcorn in the microwave as we're looking at the participants here and wait until they stop popping.
Yeah,
they will pop a little bit.
How many, Miguel? 41 in Zoom, and how many in YouTube?
In the YouTube, I have to connect in the YouTube because some people go to the YouTube. And also I have to go there because sometimes they post questions there.
Okay, they are already entering 44, now 45, 46.
So yeah, now it's time for me to ask people to say, where are you connecting from? Galera no chat, coloca de onde vocês estão conectando. Say where you are connecting from in the chat. So let's see. I saw a girl from Manaus. I see here João Paulo from Northeast, Natal, Yancas from Manaus, Campinas e São Paulo. Yeah, sometimes we have to have a map for Lars to know. Jabuticabal, São Paulo too. São Paulo is a state from
we have people from all the main 5 regions in Brazil. The north, northeast, center, west, southeast, and south.
Yes.
There's Samuel connecting from Moçambique.
Moçambique.
Rio de Janeiro, Rio Grande do Sul is the very south of Brazil.
Samuel Chumana from Mozambique.
Yeah.
Really?
Goiânia too. Please say in the chat where are you guys connecting from for us to know. And we realized that when we have a talk like yours, we have to share with different programs. Some on bioinformatics, some on genetics, evolution, whatever.
Yep.
Pitangui, Minas Gerais.
Zoology, botany, so, and of course,
of course,
your lecture. There are many people from very distinct areas.
Yes.
And PhD programs.
Here in Natal, we have the bioinformatics graduate program and also the systematic and evolution graduate program.
Systematics and evolution. Okay.
So when you say Natal, do you mean Natal in South Africa?
No.
It's Brazilian Northwest.
Okay.
Northeast of Brazil, the very east
of one of the northeastern states in Brazil.
Yes.
Miguel, put the map, please.
Sure, yeah, I will send a map to everyone who is talking because it's difficult to explain. But we have people from Amazon, up to Rio Grande do Sul. So we are covering pretty much of Brazil and many other places. So, okay, so we are going to have Dr. Lars today, and we'll cover phylogenetics, and in some minutes we'll get started here. Some people are popcorning here, but it's a pleasure to have a seminar like this, Dr. Lars, and thank you a lot for accepting. And
oh wow, brilliant!
Here's the map. So
here's the map.
Who
Yes.
you shared the map? Yeah.
Yes.
There are people from São Paulo. There is also a bioinformatics program in São Paulo.
It's a huge country. So.
It's the huge city of Brazil.
Yeah.
There is someone connecting from Germany here. See?
Yeah, I saw Bonn.
So we have Porto Alegre here, São Paulo, Rio de Janeiro, Belo Horizonte. Natal is here. In the northeastern state, Manaus in the middle of the Amazon biome. So we have people from all the 5 main regions in Brazil. To see you. I'm going to disconnect you.
Okay, the camera. Dr. Lars, would you like to try your sharing your screen to see if it's okay, working just fine? But you are used to Zoom, right?
I am, yes.
No, maybe I'll ask João Paulo to introduce you, because João Paulo uses phylogenetics a lot. João Paulo, would you like to have the honor to do the introduction today and be in the chair of this session? So please go from Natal, João Paulo.
Okay, so it's an honor to present Dr. Lars. Dr. Lars holds a master's degree in population genetics from Aarhus University in Denmark, and a PhD in molecular evolution, from La Trobe University, Australia. He carried out postdoctoral research at La Trobe University, Université d'Ottawa, Australian National University, and University of Sydney. In 2001, he was appointed to the position of professor at the University of Sydney, then promoted to senior professor in 2004 and associate professor in 2008. In 2002, he co-founded the Center for Technology and Bioinformatics at the University of Sydney. And in 2003, he was appointed its director. In 2009, he was appointed to the Commonwealth Scientific, Industrial and Research Organization of Australia to establish and lead the genomic bioinformatics research team in its division of entomology. In 2016, he was appointed honorary professor at the Australian National University, and in 2018, he was nominated visiting professor at University College Dublin. So, Dr. Lars, thank you very much for your presence here, and I am really anxious to see your lecture. Thank you very much.
Okay, thank you very much. First thing, just call me Lars. I'm not much of a person for titles, and it's not anxious, but excited. That's what you meant. Anyway, this is the first time I've given a lecture in via Zoom for quite a while, so I may be a little bit rusty. At the same time, when I get excited, I may talk a little bit fast. So what I want to do up front is that I want to try and see whether it's possible for you guys to stop me if there's something that I say that you do not understand. In all likelihood, if I say something and you don't understand it, it's because I didn't say it clearly enough. I didn't explain myself clearly enough. It is not you that is stupid or dumb. And in all likelihood, everyone else is confused about what I've said. So are you happy for, I'm happy for you to interrupt me if you want to. Is that fine with you guys?
Yes.
Fantastic.
No
Fantastic.
problem.
Okay, I'm going to tell you a little bit about a new phylogenetic protocol and how that helps us to deal with what I call model misspecification and confirmation bias in molecular phylogenetics. It's a talk that is based on a paper that I published in 2020, and it is something that is evolving a little bit further as time goes by, with different people contributing to it. So the whole idea of a new phylogenetic protocol may be mine and my co-authors, but the solutions that are needed is for everyone to develop. So let me see here. I'm just gonna, okay, yeah. So the work that I am going to present is not just done by me, but it's done by a whole lot of people. And so if you look up, I'm just going to see whether I can get my cursor here. Yep. Okay, a nice red cursor here. Oh no, let's not do that one. Use another one. I'm just going to see here whether we laser cursor, that's better. So in terms of the phylogenetic protocol, this is something that Barbara Holland and René Cotula and I have developed or have talked about for many years. The first time I thought about it was in 2007, and then it slowly began to develop. In terms of accuracy on multiple sequence alignments, this is some work that I've done with Angela McGoffrin and Simon Easdale, Charlie Robin, and a student of mine called Ramandeep Kaur, and a former student of mine, Tanya Gulucic. Completeness on multiple sequence alignments is another area, and that's with Thomas Wong, Subha Kalyanamurthy, and Karen Moisman, David Yates, and Bernie Misov. So this is also published in NAR Genetics and Bioinformatics. We need to look a little bit into multiple sequence or match pairs test of homogeneity, and that's to do with finding out how marker processes can be extracted from sequence data, and not just extracted, but whether they are the same or not. We need to look at multiple sequence alignments. This is again with a variety of people. Leon Poladian, that I highlight here, unfortunately died a few years ago from a brain tumor. Was a wonderful mathematician, applied mathematician, quick thinking person, and a real powerhouse. And unfortunately, he had a brain tumor and died at the age of 57. Then we look at phylogenetic inference under general marker models. And this is with a PhD student that was a PhD student with me and Leon. And we also incorporated a computational scientist called Michael Ott. And then there's some applied research on insect genomics, which is to some degree led by John Oakeshott with a post-PhD student of mine and some staff and colleagues that we have in different places. So when I give a lecture like this one here, I try to start out lightly. And the first question I ask myself is, we need to know what phylogenetic research is. Essentially, it's the study of evolutionary patterns and processes. So patterns here and process are two aspects of the evolution here. They underpin all extant species, including their sequences, the traits, their behaviors, and so on. So we have an example here where we look at just 5 different insects, all bees, where we have the jewel wasp, we have some bumblebees, and we have some honeybees here. And they have an evolutionary history, which is the tree-like structure that we have over here. And that tree-like structure can be inferred from genome data. And when we do so, we can actually infer how far back in time the different species diverge from one another. The pattern that I refer to above here is really the tree. And the process, the evolutionary process, is the clock-like mechanism or non-clock-like mechanism that leads to the accumulation of differences in genomic data. Why do we do phylogenetic research? Well, we do so because that knowledge that we may acquire from real data can allow us to do a lot of different things. We can do research, we can improve research, we can use it, for example, to predict gene functions. We can use it in the development of novel observations, novel products, and so on. For example, I have been involved in engineering novel enzymes, omega transaminases, where we ended up taking out an international patent on it because, or these enzymes, because they can be used in an industrial setting. That can be used in the context of management of natural resources. And many of you guys may be interested in diversity and spread of pests, or the same in diversity of spread of pathogens. So one example would be dengue fever, yellow fever, Ross River fever, Zika virus, and so on. Those are some of the examples. The type of data that is used in phylogenetic research is another question. What type of data is used? Typically, we use alignments of nucleotides or amino acids, and often these sequences are aligned, which means that we may have to insert some alignment gaps. So here's a couple of examples. These are artificial alignments. You will see that because the sequence names are essentially the names of different types of pasta. What are the challenges? In terms of phylogenetics, the challenges are in some ways quite simple, but in other ways quite complex. One of them is to infer the correct tree. We need to infer the correct tree in many cases, or we want to do so. And if we have what we call R sequences, then the problem is that there are that many different tree topologies to consider if you have R sequences. So if you have 10 sequences, then you have 2,027,025 different trees to consider. If you increase R from 11 or from 10 to 11, you end up increasing it up to some, I think, 35 million tree topologies. And that number just increases astronomically. The second problem is that for that correct tree, you want to infer, or for the correct tree that you hope to find, you want to get the edge lengths correctly inferred. The problem is that there are essentially 2n-2 edges that you need to infer the length of. And that can be quite challenging, especially from a computational point of view. Then you want to infer the correct substitution model. And the problem is that there is a large number of models and they are unknown, and you want to be able to identify what is the correct model. And also you want to infer the correct rate heterogeneity across sites model, or a RAS model. And again, the problem is that the optimal model is unknown. So they're essentially just 4 small problems. They're easy to explain, but the solutions are very, very difficult to find. So the question that arises out of this, of course, is it really possible to infer million-year-old evolutionary events from molecular data, given that these challenges here? And the answer, of course, is yes, but it becomes a challenge. It's possible to infer them, but if you want to do so in a proper manner and get the results that are reliable, results that we can have any faith in, then you need to follow certain approaches. And those approaches are described in what I call phylogenetic protocols. Here's a simple protocol. You start out by selecting your data that you want to analyze. Then you go ahead and select the alignment method. So you may use cluster W, you may use muscle, you may use a structural alignment. The choice is yours. You align the data, and then you select the sites that you want to use for the subsequent analysis. Those sites may be all sites, or you may use a subset of those sites. And then when you've done that, you may select your phylogenetic method. And when you've done so, you may use, for example, parsimony methods, or you may use model-based methods. And if you use the model-based methods, then you need to use or select some substitution models. You need to choose some marker models. And when you've done that, you go ahead and infer the phylogenetic tree. And when you've done that, you interpret the results. That is a very simple phylogenetic protocol. It's one that most people who do phylogenetics would be familiar with, and many of those people will probably have applied it. When they get to that final stage down here where we look at interpreting results, if those results are found to be consistent with our prior expectations, we tend to move ahead and write a scientific paper about it because we want to publish the results. If, on the other hand, the results are inconsistent with our prior expectations, we start using feedback loops and ask the question, is the result due to the fact that we used an inappropriate phylogenetic method? It wasn't the right method for us to use. Or did we choose the wrong sites? We may go further back and say, did we align the data correctly? Or we may have chosen the wrong alignment method, or we may even have chosen the wrong data for this approach. If I were there in person and gave this lecture here, I would ask how many people in the audience here have applied that approach here. And it's not unlikely that a very significant proportion of the audience would have applied it. The shocking thing is that that approach is not really appropriate. And it may come as a surprise to many of you too. And it certainly came as a surprise to many of the people that I worked with when I was working at CSIRO, because they were adamant that the approach that they used was fit for purpose. The reason why this approach is not appropriate is that when we get down to this stage here and we ask the question, are these results consistent with prior expectations? By doing so, we essentially use our prior expectations as a guide that helps us to determine whether the results are trustworthy and reliable and newsworthy, or they are not. That's simply not science. It's a flawed approach. We need to change that approach. We can't continue to fool ourselves, blindfold ourselves, and mislead other people who rely on phylogenetic research, that other scientists, policymakers, or even the wider public. We should not apply this phylogenetic approach. What we should do is that we should use another approach, and that is what this paper that I have the source for down here in the bottom part of the paper, that is what that paper came up with as one possible solution. I'm not saying it's the final solution, but it's certainly a step in the right direction. The approach goes like this. You start out by selecting the data for your analysis, and then you select your alignment method, and then you align your phylogenetic data, and then you select your sites for your phylogenetic analysis. And by that stage, it begins to go different. Now we have to survey the phylogenetic data. And the way we survey the phylogenetic data is that we compare the phylogenetic data to the assumptions that the phylogenetic methods are based upon. Many of you will be phylogeneticists or have done phylogenetics mainly from an applied point of view, and some of you may have developed phylogenetic methods. And those of you who are sufficiently well educated in this particular area will understand that the phylogenetic methods that I highlight here are based on some assumptions. Often those assumptions are described in the papers that describe the methods, and sometimes they are described in a manner whereby they are hard to understand what the assumptions are. nevertheless, they are there. And if the assumptions that the methods are based on are not met by the phylogenetic data that we have over here, then we have something that is called model misspecification. And when we have model misspecification in a phylogenetic analysis, or any kind of analysis for the matter, we might run into the problem that the misfit between the data and the models that we used to analyze the data, that misspecification or that misfit leads to a result that is not trustworthy, that can't be relied on, and we should try and find a better method. Unfortunately, in phylogenetics, that has been the case for the last 40 years, where we really haven't used methods to survey phylogenetic data adequately and appropriately. And for that reason, we may very well have misled the public and policymakers worldwide for many, many years. And that, in my view, needs to change. It's a controversial point of view, I admit that. But at the same time, I think it's time for us, a community, to wake up, take the bull by the horns, and actually change our behavior and our approach and do far, far better. The public deserves to know that we are changing, that we are not just using the same approach that we have used. So what happens then when we survey the data? If we go ahead at that stage here, if we find the data do not meet those phylogenetic assumptions, then we need to go back, use the feedback loops from there, go back and select other sites, align the data better, select better alignment methods, and perhaps even go back and select other data sets. Now, I'm not sure what, yes, now it's back again here. If, on the other hand, we find there's a good fit between the phylogenetic data and the assumptions of the phylogenetic methods, we go ahead and select the phylogenetic method. We use the same approach as we've done before, get to the stage where we infer the phylogeny. And at that stage, we now need to use a parametric bootstrap approach. We need to do so because we need to test whether the fit between the phylogenetic data, the phylogenetic models that we use, or the marker models that we're using, and the phylogenetic result is good or not. That is, we can use that in a likelihood context. We use a parametric bootstrap analysis. We could do the same in a Bayesian context, in which case we are using a posterior probability, predictive probability approach. And if it turns out at that stage that we have a poor fit between tree, model, and data, then we go ahead and use this, follow the feedback loops, select different marker models, different phylogenetic methods, or we may to go ahead and survey, or go all the way back and survey the data using better methods. If, on the other hand, we have a good fit, we can go ahead and interpret the results. This is a subtle difference, but an important difference in the phylogenetic protocol. And it's the one that I want to leave all of you guys with as probably the most important aspects of this particular lecture here. At this point here, I just want to stop at this stage here. So this is the lecture, or this is the new phylogenetic protocol as it looks like here. And I just want to stop and go back and see whether there are any questions in the chat. This is the time for you guys to enter the chat and go in and give me some questions if you have any questions in that. So can I see whether there are any questions at that stage here?
Not yet.
Not yet. Well, I can see there are 3 new messages, but none. Okay, fine. That's one here. And Hola, Pessoa. I don't speak Portuguese, so I don't really know what it means.
Just the presence list.
Yeah. Okay, yeah. Shall I continue?
Yes, for sure.
Okay, good.
Yeah.
I will do that. Go back to the slide here. Where are we? There we are. Share screen. Okay, back to share. Why is it not going? Oh, yeah. Here we are. Just bear with me. While I get that. No, why is it not? Okay. This is really annoying me a little bit. Okay. Something is not. Oh, yeah, there we are. Good. Okay. Now we are back to the slide. Okay.
Okay.
Great. Okay. Okay. So looking at this new phylogenetic protocol here, I have worked in a number of different areas, focusing on either selecting alignment methods, selecting sites for analysis, surveying phylogenetic data, selecting phylogenetic methods, working on model-based phylogenetic methods, mainly in the likelihood area. Marker models, model selection is a big thing in my area of research, inferring phylogeny and the parametric bootstrap analysis. So I'm going to, in the rest of the lecture here, focus on some of the work that I have done in these particular areas here. So, okay, yeah, I'm going to first focus on the multiple sequence alignment areas. Many of you will have focused on multiple sequence alignments and will know that the objective is to obtain a matrix of R rows and C columns, where the rows represent the sequences in your data and the columns represent the homologous characters, typically nucleotides or amino acids, sometimes even codons. The problem is to infer an accurate multiple sequence alignment is in fact what I would call a non-trivial task. And that is because the alignment methods focus on similarity, and that similarity you have between sequences in alignment is due partly to the homology and partly due to analogy, where homology refers to similarity due to historical relationships that occurred by descent. And the analogy is due to similarity that's due to all other causes, common functions, etc., etc. If you want to know more about this, then David Morrison has written an excellent paper on this in a journal called Systematic Botany, volume 14, page 14 to 26. You may not be familiar with it. It's an excellent read. Now, the issue that I was faced with back in the noughties and even early on, because the very first time I discovered this issue here that I'm going to show you was actually in 1997. But I never really published about it before I got a postdoc who came in and worked for me for a couple of months, Tanya Golucic, who is a delightful person to work with, an incredibly intelligent person. And she has worked in the UK since for many years and has just returned back to Australia now to work in an academic position at the University of Sydney. As far as I know, it's Sydney. What we did was that we chose to take data. Here you have an alignment, a reference alignment, where all the sequences are identical to one another, except for the gap. Here I have cut away some residues, some amino acids, in all the sequences except for the first one. So the sequences are, for all intents and purposes, identical to each other, except for where the gap is. Such sequences should be trivial to align by any alignment method. You can see they align very, very well. On the other hand, if you take such data and try and align it, you get different results. Here's an example of the world-famous cluster W alignment method that Des Higgins and Toby Gibson generated and published in 1994. So Des Higgins is a friend of mine. He is actually, his office is just over there, or rather, that is his past office, because he has just retired from the University of College Dublin and is now spending his time doing other very, very interesting things. He was rather shocked when he saw this alignment here, because he never thought that was the case. It's not just cluster W that gets has that particular problem. MAFT, PropCons, and MOSL, which are other multiple sequence alignments that many of you will have used, especially MAFT, I suspect, and especially MOSL, and probably also cluster omega, which is the sequel to this one here. What you find is that there is a propensity for these alignment methods to align the gaps rather than aligning the residues. In other words, in this particular case here, the gaps take precedence, aligning the gaps takes precedence over aligning the residues correctly. That's a big problem. Some people, when they saw this and reviewed this manuscript here, said, well, that's a very construed example. Nobody would have questions or have to do something like you suggest doing here. And we countered back and said, well, you're wrong. As a matter of fact, if you look at alternatively spliced gene products, you will find many, many cases of that situation where you are trying to align sequences that are identical, except for where the gaps are. And so we incorporated a couple of examples of that and found that that was in fact the case, that these alternatively spliced gene products also are incorrectly aligned. So that was published in 2007. And then we went ahead and Angela McGoffrin, who was a postdoc of mine, is now a senior lecturer in New Zealand, did an analysis where we looked at another example. We got it very far, the analysis got it very far, and we're writing up the manuscript, and then I discovered that there was a bug in some of the software that she had developed for analyzing the data, meaning that the whole analysis had to be redone. And so I spent several months last summer, reanalyzing the data, and we're still in the process of summarizing the results. And I'm going to give you some example of it here. In this case here, the question that we asked was, how do popular multiple sequence alignments or methods perform when alignment gaps are not necessary? That is, if you simulate the evolution of a nucleotide sequence or an amino acid sequence on a tree, and you do not insert any gaps or introduce gaps or deletions or insertions during the evolution of these synthetic data, you end up getting an alignment that is correct. And then you can apply the multiple sequence alignment methods to those alignments. And then you can look at how often the alignment gaps are introduced. That gives you an insight into how accurate these alignment methods are. So we did that, and we surveyed multiple sequence alignments for evidence of gaps in sequences with 1,000 sites generated on different trees with 16 tips. And we surveyed, as a matter of fact, 20 different multiple sequence alignment methods. And I'm going to give you the example or the results from one of them. In this case here, we looked at, I can't remember which one it is. This might be cluster, yeah, this is cluster omega that we used here. What we have in the first panel here is the length of the tree measured in substitutions per site. So if you have a tree with 16 tips, you can add all the edges together. And when you do that, you get what is called tree length. And that tree length tells you how far or how much the data has evolved over the tree that you are considering. And so if you have a tree with tree length 5, you have a lot of changes occurring, but all in all, over the whole tree, that variation that you have generated corresponds to roughly 5 substitutions per site over the whole tree. Up along the y-axis, you have a value, a parameter that's called AEL, which stands for alignment extension length. That is essentially a parameter that says if you have an AEL of 0, then your alignment is 1,000 amino acids long, or 1,000 sites long. If AEL increases, it means that you have the alignment has some alignment gaps introduced. And because of the experimental design, you would not expect any of those alignment gaps to be introduced. So if they are introduced, you know that there's a problem with the alignment method, that the alignment method introduces gaps that are not needed. Now, along the y-axis, we have values from 5 to 40. Now, 40 substitutions per site may sound rather excessive from your point of view, and I completely agree. Anyone would think so. Now, look at the figure here. When we have 5, in most cases, we just have 0 substitutions per site, but there are cases indicated by that round circle where you have more than one alignment gap. It's not many cases. They're up to about 15 substitutions per site, you are fine. But then when you have 20 substitutions per site, you begin to have a problem. You begin to see that there's quite a lot of observations where you have alignment gaps introduced that shouldn't have been there. As you go to 25, it increases. And now it's actually quite rare to have 0 substitutions per site. In some cases, you even have a doubling of the alignment length. And when you get to 30, you have none. Every single alignment has alignment gaps in them. And you have cases where you have more than a doubling of the length of the alignment. And when you get up to 40, it's even worse. That is, from my point of view, a big call for concern. And that was indicated early on when Angela did her first analysis that we had a real big problem with some alignment methods. Now, the next question we did was, we then asked the question, well, what could happen if we used the alignment in inferred phylogenetic trees from them? In this case here, we did a phylogenetic analysis using the model that was used to generate the data. And then we inferred the phylogenetic tree from each of these 100 alignments that we generated from each tree length here. And if you follow here up along the top line here, that is the result that you get when you infer the phylogeny from the true alignment, the alignment that does not have any of the gaps in the data. In this case here, you get the correct tree every single time, except for when you have 35 or 40 substitutions per site. Here you have correct trees occurred in 98% of the times and 99% of the times. On the other hand, if you inferred the phylogenetic tree from the alignment that you had inferred, in this case here using clusteral omega, you found that you got the correct tree in many cases here up till an alignment, a tree length of about 20. And then you had a massive drop in your success rate. And when you got down to about 35 and 40, none of the trees were correct. In other words, we have a very worrying observation, and that is that multiple sequence alignment methods introduce gaps where no gaps are needed, and will introduce more gaps depending on the amount of divergence that exists between the sequences. And we have no idea that that occurred, meaning that multiple sequence alignments from many published data sets might actually be misleading. Sometimes not very much, because the tree length is maybe perhaps short, but in other cases, it could be a very substantial bias that the multiple sequence alignment introduce by simply introducing alignment gaps that are not necessary. So this is just with one multiple sequence alignment method. We did the same analysis with 19 other multiple sequence alignment methods and got different results. But the big picture is exactly like this one here. And it was with different tree topologies and with different substitution models, etc. So it's a very, very clear indication that we have a very big and so far unrealized problem with multiple sequence alignment methods that needs to be resolved. Okay, enough about this. I'm now going to, should I stop and just take questions at this point here, or Miguel, if you just let me know whether I should continue or just wait.
We have one observation.
I have a comment.
Yes, please.
It seems like a phase transition, a clear phase transition in that graph between the 20 to 25 substitutions per site.
Yes.
It's a phase transition, and this is, at least in physics and chemistry, this is the most interesting thing when you are studying a system, a physical, chemical, or even biological system. So it's very interesting because phase transitions are when one has the most interesting features to retrieve from a system. Mm-hmm. That's very, very interesting. Very interesting.
It's not just interesting, it's also extremely worrying from
yes, of course.
From my point of view, as an evolutionary biologist, you know, when you think of how many resources have been put into inferring phylogenetic trees, and where many of those most of those analyses rely on multiple sequence alignment methods. What I suspect, my instinct is that we need to go back to the drawing board, and we need to actually start developing new multiple sequence alignment methods, and that require a very substantial input, possibly from physicists who understand this much better than I do as a biologist.
This is a typical phase transition. Typical. And that's when we study any system. This is the aspect that everybody is looking for, but for a good thing, not a very worried thing in the case of phylogenetics. But fantastic.
Okay. Any other comments?
Yes, please. How did the I'm just curious, how did the programs handle the gaps? No gaps at all, or they just excluded the gaps? Just as curiosity. Okay,
you mean the phylogenetic methods in this case, because the alignment methods are introducing the gaps, right?
Yeah. But
so the phylogenetic methods, when a phylogenetic method identify a gap in an alignment, it will iterate over all possible states that gap could be in. So if it's an amino acid, a data set of amino acids, you will integrate over all 20 possible states in that position, which of course causes the methods to slow down and probably also increase the variance a little bit.
Thank you.
Other questions?
Lars, I'm wondering here. I understand that from your examples, the gaps are introduced by error there, But I'm wondering if all the methods, when they do the next step, that's to measure the distance.
Yeah.
If they get a good result, so they don't even look at this problem that the alignment has.
Yeah. What they do is that when they measure the distance, that would be more like when you do a distance-based phylogenetic approach, where you take sequences in a pairwise fashion, compute a distance, put that distance into a matrix, and then use a clustering method to convert the distance matrix into a bifurcating tree. When you do maximum likelihood analysis or Bayesian analysis, what you're doing is you take all the data and you're trying to fit all the data to a tree topology where you optimize the substitution rates, the rate heterogeneity across sites rates, you optimize the edge length, all the parameters that you build into your model of sequence evolution. All those are being optimized using, some kind of algorithm that is embedded in the software. And so you don't necessarily exclude the sites that are that have gaps, unless you go ahead and use the next step in the talk here that I'm actually going to focus on, where you are masking sites. So the question is good, and it actually leads directly into the next step.
Okay.
Any other questions, or shall I carry on?
I have a question, Lars.
Go ahead.
How do, I mean, after a while, all that's left is noise because it randomizes the data if you have too much mutation.
Correct.
So, do you know if there's still phylogenetic noise, phylogenetic signal, sorry, here left after 25, 30, maybe no alignment method will have a chance to recover things?
That is exactly why I did the phylogenetic analysis with the data where there are no gaps in the data. So you look at the black dots here, right? So the fact that I recover the correct tree here 100 times, 98 times, 99 times out of 100 means that there's still a very clear signal in the alignment that supports the notion that I have not lost the historical signal in the data when I have 35 to 40 substitutions per site in these data here.
Perfect, thanks. Yeah, I was focusing on the alignment and I didn't pay attention to that part. Good.
Yeah. No, it's a beautiful question. It's a very good question. It highlights exactly one of the criticisms that I want to avoid getting if I just present figure A here. If I present figure B, you get a very different result that you can't reject that, well, this is a serious problem. So anyway, let's go ahead now and look at the next step here, where we are focusing on selection of sites. So I want you to just consider these two silly alignments here, alignment A and alignment B. They are obviously different. And when you look at those alignments, one question that could arise in some people's minds, at least in mine, is how do we compare at multiple sequence alignments, and what metrics should we be using? I'm just going to take a drink here. The answer to that is that we could do that by quantifying the completeness of multiple sequence alignments. To do that, I want you to consider just a simpler multiple sequence alignment with R sequences and C sites. And then if we do that, we can define, for example, a completeness score for the whole alignment, which is, I call it CA here, where the subscript here means all here. And X here is essentially just the number of cells that you have in your alignment that contain what I call unambiguous characters. So for DNA, it's ACGT. And N would be an ambiguous character. ACGT are unambiguous. R, Y are also ambiguous characters. And so if you calculate that number from your alignment and divide it by the product of R and C here, you get a value here. And that value here falls between 0 and 1, where 1 means that you have a complete alignment, what everyone dreams about, and hardly anyone has, and 0, which is a non-alignment. You know, there is nothing. Some people will be familiar with this metric here because it's a little bit like the percentage of missing data that people have reported occasionally in papers. The other completeness score one could come up with is the completeness score for a sequence. So if you have your multiple sequence alignment here, then CR here, where the subscript here means the number of sites in a sequence or in a row, then it's just the number of sites with unambiguous characters in the sequence divided by the length of the alignment. Again, the value is between 0 and 1. Completeness score for the site. Now you're looking at every site one at a time here. And then XC here is the number of rows at a given site that contain unambiguous characters at the site. And then you have the perhaps a little bit more complex one to fathom, and that is the completeness score for sequence I and J, where Xij now here is the number of homologous sites with unambiguous characters in sequence I and sequence J. So that is, if you just look at the pairwise alignment, then you're looking at how many of those sites in those two sequences contain ambiguous characters in both sequences. Those are 4 very simple completeness scores, and they have a certain purpose, at least from our perspective here. So I have an example here. Now we have just a simple example from bumblebee genomes, where I was involved in the bumblebee genome paper. And we looked at, in this case here, carboxylcholine esterases. We had about 143 proteins, and the alignment contained about 2,320 sites. We used a method that's called ALISTAT. This is a software, a bioinformatics application that we developed. You can find the URL to that in this particular paper here that's listed at the bottom in NAR Genomics and Bioinformatics 2, and so on. Now, if you do that, then use that method, you can get a histogram of the completeness scores for the rows. And what you find here is that it's quite skewed towards the lower end of the range here, with no values up in the high end here, and some values down here hovering around about 0 or a little bit above 0. That means that none of the sequences are covered completely across the full alignment of 2,320 sites. We could go ahead and look at the distribution of CC scores, and that is now looking at the completeness score for the individual columns. And in this case here, we get this nice U-shaped distribution with a substantial number of sites that have completeness scores between 100 and 0.95. That's roughly speaking about 300 and 320 or 340 sites. And then you see that it drops very quickly down here to this level here, and then it slowly increases again, implying that there's a very, very large number of sites, probably in the order of about 900 sites, where there's only just 1 or 2 sequences that have residues in this alignment. This means that it could possibly be a useful approach to survey alignments and say, well, okay, if I'm going to do a phylogenetic analysis, I could say that I'm going to only use the sites in the alignment that have completeness scores above, say, 0.65 or 0.7 or 0.75, meaning I'm focusing on the part of the alignment that is well represented by amino acids in most of the sequences, whereas the large number of sites that contain very, very few residues in most sequences, very few or any at all, can be ignored. That's a possibility. We're going to look back into that in a minute. So we can also look at the heat map of the CIJ score here. And if you do that, what you'll find, the values here go from 1 to 0. And it's clear that there are cases where we have essentially 0. There's a couple of cases up here where we have 0. And if we go ahead and look in more detail, we can look at those and find out that, well, in those cases, those 2 zeros there must be due to comparisons evolving the Bombus impatiens and Bombus terrestris H-clade sequences in relation to some other sequences that we have here. Oops, that is not what I want. I just want to go back to, there's my, now I'm just going to go back to my cursor. Here we are again. We can go further ahead and look at the other part down here. And in this case here, we have green pixels. And that means that we have a very good coverage from Apis mellifera, Apis florea, Bombus impatiens, Bombus terrestris, and even Asonia vitripennis. And that's pertaining to a clade of sequences that are called L1. You can see here L2 is also reasonably well represented. So that means that the heat map actually helps us to identify subsets of sequences that could be considered having a very, very good coverage, as well as those that have very, very poor coverage. Now, I'm going to go back to the previous issue about the columns of the complete score for the column. If you want to analyze a set of data, an alignment, you often might want to use a program that's called gBlocks. The gBlocks is designed to identify sites that are considered to be too variable or to have too many gaps in them, and therefore those sites are ignored from the alignment, leaving the rest available for phylogenetic analysis. If I took these 12 sequences here and applied gBlocks to it, then I would find that those sites here are the ones that are ejected or removed by gBlocks. What you can see here is that gBlocks will actually move sites, some sites that are constant. There are no changes occurring in that column here. In this site here, there are basically 2 different residues, 2 different amino acids, histidine and tyrosine. Those are removed. Serine here and threonine again removed, tyrosine and phenylalanine also removed. And over here you have some sites with gaps in, they're also removed. Now, if we were to, that's not really useful, or that's not really desirable, because it means that we are removing sites that are well conserved in the data, and we also remove sites that are well aligned. This site here is very well aligned within the alignment. Nobody can question that particular site being there. And it's a site with 8 different amino acids. So yes, it is a fast evolving site, but it may actually still be a phylogenetically informative site. It's a site that is very good for a phylogenetic analysis. If we go ahead and mask the data using Alistat, we could, for example, say now we're going to use a threshold of where the CC score is 60% and above. And if you were to do that, then we would remove those sites here, and we would keep these extra sites here in the alignment. That means that we have more sites in the alignment. It also means that it is upon the user to choose the threshold that is used when they decide to mask the alignment. It means that in this case here, that we get more sites in the phylogenetic analysis, which is important. It also means that some of the sites that are included actually are included actually are quite variable and therefore quite useful from a phylogenetic perspective, whereas many of the sites that were included over here are invariant. They haven't changed. These ones here too. So over here, we only really have 3 sites that changed, whereas over here we have 3 as well as another 4 here. So we increased the number of sites that show evidence of having changed. And that's an important consideration to have when you do a phylogenetic analysis. So the final thing about this approach that we implemented in Alstat is that it actually increases the transparency of the research that's being done when that research involves multiple sequence alignments. We can use what is called a minimum reporting standard. We came up with this idea of a minimum reporting standard for multiple sequence alignments because we believe that phylogenetic research represents or do not describe the multiple sequence alignments in sufficient amount of detail. And so people reading the papers are not in a position where they can actually make an educated decision about the usefulness and accuracy of the phylogenetic data or the phylogenetic research that's being done. And so a minimum reporting standard is essentially just a simple alignment or simple matrix that tells you, or table that tells you the size of the alignment, the completeness score of the alignment, the maximum and the minimum value of the CR values, the maximum and minimum of the CC values, and the maximum and minimum of the CIJ values. In this case here, before we applied a threshold of 80% here, the alignment was 143 sequences and 2,320 sites. And here are the values below here. You can see CIJ minimum is 0. The CC min is 0.07%. So we have some very low values there. We also have some good, relatively high values here in some of the cases. After applying this threshold here, we were left with an alignment with 489 sites. And you can see a very rapid or marked increase in the CAA score from, I should just go back, sorry, I forgot this one here, we need to have the laser. A very substantial increase from 28% to about 95% of the completeness score. Some sequences now have a CR value of 1. The smallest CR is still only about 5.7%. The CC score is still high, and the C min score is now also very substantial. So you end up getting a very, very different alignment if you apply this approach here. And it means that you can report in your paper what the alignment was before you applied the Alstat and what the alignment was afterwards. In other words, you now have some kind of automation as well as means of reporting the result of your automation to the readers. It's a simple, but it's a very, very useful approach, at least in our view. Okay, enough about that. Now I'm going to focus a little bit more about surveying phylogenetic data. And for that, we need to understand a little bit about phylogenetic assumptions. So I'm going to go through that a little bit faster.
Lars,
because yeah.
Excuse me.
Yeah.
We have 2 questions if you want to answer, because it's about the alignment.
Okay, good. We can do that. What I just want to know is, I'm mindful of time, that it's already 6, and I have actually gone over time already, which is not ideal, but on the other hand, maybe you guys have actually got quite a bit more out of it. Up to you. Maybe we should go first back to the questions about the alignments. How far should I go back? To this stage here?
Just 2 questions on the charts. If you can see, one question from Santarelli and other questions from Ari. But we can see the questions on the
I'm just going to go back here. Maybe you actually, if you read the questions up for me, that would be
problem. Good. R. Santarelli asked, how different can be the result if the forced alignment does not occur? And what is the frequency of natural alignment without using gaps or position adjustment by algorithm?
So there are two questions in that one. Can you give me the first question?
Yeah, how different can be the result if the forced alignment does not occur?
Okay, I don't know the answer to that. It's probably very data dependent. What I do know is that when I applied these two G blocks to, for example, the acetylcholine esterases, if I applied G blocks to that one, it removed every single site from the alignment. There was nothing left. When I masked the acetylcholine esterases using a threshold of 0.8, I got 489 sites. When I then sat down with a biochemist who had worked his whole life on this particular family of enzymes, there were only 3 different sites where his decision and my decision based on allostat differed. And that was a complete eye-opener from my point of view. Of course, there's an element of luck in it and serendipity, but it made me realize just how useful allostat might be, because it means that the user can make that decision himself or herself what the threshold should be. The great advantage of allostat too is that when you use allostat and use masking with a threshold of 0.6 or 0.8, allostat will output 2 different alignments. It has the original alignment. It will output the masked alignment with the sites that are included in the masked alignment, and will output also an alignment with the sites that are discarded. And that means that you can actually take those 2 alignments and you can do whatever analysis you with either of those 2 alignments and see what the effect of changing your threshold from, say, 0.6 to 0.7 to 0.8, what effects does it have on your phylogenetic estimates? And therein lies a great advantage, because it empowers any investigator. And I firmly believe that we need to empower all the users that use software, bioinformatics tools, make it possible for them to apply common sound knowledge and use that in this, using their bioinformatics applications without using what I would call black box approaches, where you do not really know what goes underneath the bonnet or inside the program. So that was answering the first question. Then there was a second question.
Yeah, there is the second question, but he also said that the slides 15 to 18 give me the answer of the second questions.
Yeah, give me that second part of that first question. It was?
The first question and the other second questions. And what is the frequency of natural alignment without using gaps or position adjustments by algorithm?
I haven't got the answer to that either. What I can say is, in my research going back to probably 95 and to now, it is very, very rare that I see a multiple sequence alignment that does not contain some gaps. We published earlier this year a reference alignment for mitochondrial genomes from humans. And when I started this work, I aligned 8,000 mitochondrial genomes from humans. And many parts of those alignments were, or those data, or that alignment, were in incredibly good condition, but there were always some areas where the data did not align, where there were certain insertions and deletions. That's for a mitochondrial genome. For nuclear genomes, it's the same issue. When you look at nuclear genomes of mammals, I have not yet come across a gene family where there isn't a gap somewhere in the alignment. So I think gaps are, they are here to stay. They are a natural component of multiple sequence alignments.
Thank you, Lars. You may continue now. We're going to take questions to the end of presentations to the presentation stay more fluid, right?
Okay. Well, I like the questions actually. So, and I feel they are really good because when you speak as a speaker, if you continue just speaking and telling, you don't actually have an opportunity to see what the reaction is in the audience. Remember, I can't see you guys, see your reactions. You might have fallen asleep perfectly fine by me, even if you were present. So anyway, do continue to ask questions if you feel like it. And stop me if you feel that we have reached the end of time. Is that fine?
Okay, no problem.
Good. Okay, let's look at the survey of phylogenetic data. I want now to go to the phylogenetic assumptions. And if you think you have a look at a multiple sequence alignment, then most phylogenetic methods will assume, some very simple assumptions. One is that the evolutionary process at each site is what we call Markovian. That is, it's a memoryless process. We also assume that the evolutionary process at different sites are independent of each other and that they are identically distributed. That means that the probability of change from A to C and A to G is the same across all the sites. There might be some variation, and that is that some sites may evolve a little bit faster than others, and that can easily be modeled using a simple approach here. And then finally, we assume that the evolutionary processes are what we call stationary, reversible, time homogeneous. That means, in other words, if we have a multiple sequence alignment looking like this little one here, 3 sequences, those 3 sequences are listed up here at the very top above the red line. And below the red line is all what we want to infer. We want to infer the evolutionary process. We want to infer the tree topology, whether it's this one here or whether it's one of the other ones. We sometimes may want to infer what the ancestral sequences are. And we want to find out whether the process here is the same as that one here or whether it's a different process. Now, the phylogenetic assumptions, the last one here about the stationarity and reversibility and time homogeneity, is one that's particularly important from my point of view, has been for many years. Now, a process, a Markovian process, can be what you call stationary. It can be reversible, and it can be time homogeneous. That's a possibility. There are other possibilities here. And of those 8 scenarios or processes here, 2 of those, number 2 and number 6, they are not possible. In other words, you can't have a non-stationary process that is reversible. It's not a possibility. It's mathematically impossible. And the same one with this one here. Now, that is essentially a process over each individual age. Now you have to go beyond that and say, well, okay, it's not just each individual age, it's over the whole tree. And therein lies the problem. Now, the phylogenetic methods that most of us use, and many of you probably will be familiar with, actually assume that the process is stationary, reversible, and homogeneous, time homogeneous, over not just individual ages, but over the whole tree. In some cases, there are phylogenetic methods that assume that the process is stationary but not reversible, and it's time homogeneous. Those methods include the Lee marker models that Matthew Woodshams and others from Tasmania in Australia published a paper on in Systematic Biology many years ago. You also have cases where the processes are non-stationary and non-reversible, but they're still time homogeneous. They are also possible. There is, however, situations where sequences may have evolved under stationary and reversible, but not time homogeneous conditions. And scenarios 7 and 8 are also possible. What I'm going to argue here in the rest of my talk is that we actually need to bear in mind that scenario 5, 7, and 8 may actually be the more realistic scenarios that we have occurring when we look at DNA and protein. And we need to develop methods and use these methods when we analyze the data. Now, to understand a little bit more about this here, this was just from a single model. Now look at the model of sequence evolution over the whole tree. We have a very simple scenario, and scenario 1 or process 1 that I showed you in the table beforehand essentially is one where you have the same process over each individual age in the whole tree. It's a very simple one. The most complex one is where you actually have a different model over each individual age in your tree. Those processes are different. The consequences or implications here are very different. If you look at the first one, then essentially, if the process has been like this one here, then compositional heterogeneity across sequences is unlikely to arise. That is, if you look at all the sequences that evolve over this tree here and reach this tip here, these tips here, then those sequences will have very, very similar nucleotide composition or very similar amino acid composition. On the other hand, the implication of this over here is that compositional heterogeneity across the sequences may develop. You can flip that one around, that argument around, such that if you find compositional heterogeneity across the sequences, then it's very unlikely that this scenario, the simple model of sequence evolution, actually occurred. It's more likely that the model of sequence evolution is complex. The question, of course, is how do we deal with this? I just want to show you this example here, because here's an example from a paper that we published in 2004, where we did this experiment. We simulated data on a 40 tree, and we started out with an ancestral sequence here 100 million years ago, and then that sequence diverged into 2 ages here, and the length of those 2 ages here correspond to 1, 2.5, and 5 million years ago, or later. And then we had 2 divergence events occurring again. And at that point here, the sequences began to evolve under different Markovian conditions, with these 2 sequences, A and D, acquiring higher GC content, and B and C acquiring smaller GC content. Then we inferred the phylogeny of the data and looked at the success rate. Out along the y-axis, you have the difference in GC content between C and D and A and B. And up along the y-axis, we had the frequency of correctly recovered trees. And in this case, we used maximum parsimony. What we found here is that the success rate was quite good for the tree with 5 million years between the 2 divergence events. And then eventually it dropped down here to 0. If we changed the length of that internal age and increased the length of the terminal ages here, we found that that change from 100% to 0% started early on and finished early on. And when we were down to 1 million year difference between these 2 nodes here, we found that that actually even started here at about 80% and dropped down to 0% even earlier. In other words, if you look at sequences and you have compositional differences among them, then it's pretty clear that you are very likely to have data that have evolved under complex heterogeneous conditions or complex Markovian conditions. And the difference in the nucleotide content or amino acid content that you detect is not sufficient to allow you to say something about these data have not diverged far enough to be of any concern, because you don't know the length of the true internal edges. It could be that that true internal edge is very long, in which case it's fine. You won't have a problem with the compositional heterogeneity if it's up here. On the other hand, if the true internal edge is very short, well, then you may actually be down here or all the way down here. In other words, be careful. The problem is that, well, how do we assess this for real data? There are some methods that are published, and they have already been published, and they can be used before doing the phylogenetic analysis, and they are attractive for that very reason. There are others that can be used after phylogenetic analysis. They are more accurate, but it means that you may waste a lot of time doing a phylogenetic analysis before you can get the results. And then there are some problems with these here. Some of these methods have poor statistical basis, some require a tree in order to work, and some cannot easily deal with very large data sets. For this reason, we decided to focus on some methods that examine the homogeneity of what you call divergence matrices. You want to look at the pattern of symmetry or lack thereof in these divergence matrices. In order to understand what I mean, look at this simple two-tip tree. You have a sequence that evolve along the tree, get up to this point here. Bifurcation, you have two identical sequences. And if you look at your divergence matrix, you will have 294 sites with A in both sequences, 372 sites with a C in the same sequence, or in both sequences, and so on. All the off-diagonal elements are 0, meaning that the sequences are identical. Now, if we allow the sequences to evolve further up here, then you will see here that the divergence matrix has changed, and now you have indication of some variation in the data. What you are interested in is you're interested in comparing this value to this one here. That is, how many sites do you have A in one sequence and C in the other, versus how many sites do you have C in the first sequence and A in the second. You compare matching sites here, matching cells in your divergence matrix. And when you do that, you have a certain framework set of statistical tests that are applicable. These are called matched pairs tests of homogeneity. The first one here is by Alan Bowker from 1948. The other one is by Stewart from 1955, matched pairs test of marginal symmetry, where you look at the marginal values here. And then you have the final one, matched pairs test of internal symmetry, which was written by my PhD student and the colleagues, the collaborators that we did this particular work with. Now, these tests here have different test statistics and different degrees of freedom, and they have a certain relationship. That is, the test statistic from Bowker is equal to the sum of the test statistic from the other two tests. Now, how does this work? Here's an example from a simple analysis where we look at sequences that evolve under stationary and reversible conditions. In this case, you will see here all the dots in this plot here, which maps the expected probability and the observed probability from the tests against each other. They fall down along a nice diagonal here in the PP plot. These are called PP plots. If you, on the other hand, have that the sequence process was stationary but not reversible, well, what you find here is that Bowker's tests give a very different result. Stewart's test of marginal symmetry have the dots falling down along the diagonal, and Abamnet's test have a plot where they fall again down along the bottom. If you have non-stationary and reversible processes, you get a different result. In this case, it's Baugers and Stewart's that give rise to the clear deviations from your expectation, whereas Abernethy's test is pretty much similar to this one over there. And over here, we have a situation where if the evolutionary process is non-stationary and non-reversible, well, then you get these all three tests showing this particular product. In other words, the tests that we have here actually are quite useful for detecting different types of evolutionary processes. They can distinguish the different types of processes. Now, those of you who are familiar with statistics might have noticed that in the previous case here, these were just individual tests repeated again and again and again, which is unrealistic. The more realistic scenario is that you actually have a set of sequences that have evolved over a particular tree, in which case the data, you're doing multiple tests between this one here and that one, and then this one and that one, and so on. And the tests of the sequences are not independent of each other anymore, because there is an underlying tree, namely the tree you have over here. So we did some simulations on this. In this case here, we just took this 22-tip tree over here on the left and allowed the sequences to evolve under complex, but otherwise globally stationary, reversible, and homogeneous conditions. And then we got this result here, where you find that again here, the PP plots show that the p-values fall nicely along the diagonal in every single case. You can also look at the data using what we call heat maps, which map the probability from your test from each individual comparison in your heat map. And if you do so, what you find here, in this case here, you get some nice heat maps with fairly lightly colored pixels in it. Now let's have a look at a mammalian genome dataset. In this case here, from a paper I co-authored in Nature, Webb et al. We have 48 species here, and we had about over 21 million sites. In this case here, from 12,913 protein coding genes. We used a program that's called SimTest, and here's what you got from your first codon site. Here's what you got from your second codon site, and here's what you got from the third codon site. In other words, you can very, very clearly see that these sequences have not evolved under stationary, reversible, and homogeneous conditions. We have probably a scenario where it's the worst possible situation we could be in. And this is, I'm sad to say, unfortunately, often the case for other data sets that I've looked at. We could go ahead further with this one here, and that's exactly what we did in that Nature paper, and that is that we surveyed each individual protein coding gene, and we looked at each individual codon site in it. And when we did that, we ended up with a data set where we ended up having just 488 protein coding genes here. And now we have a very, very different result. We still find, in this case here with the Stewart's test here, that there's a very distinct group here of sequences where these ones here, which I can't quite see what the species names are. Sorix is one of them, but they seem to have evolved under certain conditions that are different to many of the other ones evolving under other conditions. So it's very clear that we have now an approach that tells us that these sequences have evolved under non-stationary conditions. If you look at Abernethy's test, there's only really one sequence that stands out, and that's this one up here, meaning that most of the other sequences have evolved under what looks like reversible conditions. That's a great advancement because it means that we could go ahead and use these 288 protein coding genes and analyze those. That is essentially what we did in that paper. I'm just going to stop here and find out whether are you interested in me to continue, or shall I stop? Anyone there?
You may continue.
Okay, I will continue.
Yeah.
Okay. Okay, so now I'm going to focus a little bit more on the selection of marker models. So I'm going to go there. So I want you to think about how we model rate heterogeneity across sites, or what I call RAS. There's a number of common approaches that all are within what I call homotacous models. I'll explain what homotachy means in a minute. So one approach is to assume that there's a certain proportion, P, of sites that are what we call invariable. There's another approach where you model heterogeneity across sites using a discrete gamma distribution with K rate categories, and where there's a shape parameter that shapes that gamma distribution. Then that if you do that, well, what you have is that there's an implied assumption that all the rates, the ith rate here is essentially larger than 0, and that the probability of belonging to the ith rate category is 1/k, and that the sum of all the products of pi and ri here equals 1. You can go further ahead and you can actually combine I and gamma here. And if you do that, well, then you have several parameters here to think of. One is the proportion of invariable sites, your shape parameter, and the number of rate categories. And then you can incorporate a new approach that we introduced into the model selection domain. It's not us who is the inventor of this new method here. That is actually Seeing Yang from UK, who's very, very well known by most people in the phylogenetic domain. That is what we call a free rate model, or a probability distribution free model. It basically have these three parameters here, and it means these have these properties here where the rate of the ith rate category is larger than 0. The probability of belonging to rate category I is equal to a value between 0 and 1. The sum of these probabilities equals to 1, and the sum of the products of these values equal to 1. The value of this particular model here is that it's far more free. You can have rate heterogeneity across sites that are far more complex in their appearance. They can be bimodal, trimodal, you name it. They're all what we call homotacous, though, and that means that if a site falls into a given rate category, then every edge in the tree will evolve at the same rate. And that may not necessarily be realistic. We implemented this approach, the new R model, in what we call Model Finder, and we published it in a paper in Nature Methods in 2017. And we had a look in this case here. I'll just illustrate how accurate the method is. In this case, we took a data set from Wu et al. Tree of Life data that had 353 sequences and about 6,597 amino acid sites, all from microbes. And then we used Model Finder, which is embedded in IQ3 and subsequent versions of it. What we have out here is a complex figure. The paper, Wu et al. Analyzed the data using the WAG plus I plus gamma 5. Don't know why they used gamma 5. And what they did was they got a BIC score, a Bayesian information criteria, of this particular value here. And then they published the tree. When we used Model Finder, we actually found the LGR14 was optimal. And the difference between that optimal model and that inferior model that was published was somewhere in the order of, I think, 30,000. I didn't actually write. No, there's 30,000 here, and there's 10,000 there. So we have about 41,600 and so odd, 94 difference in the BIC score, which is absolutely huge, especially when you think that a BIC score difference of about 10 is considered very significant. We went ahead and looked a little bit more at other models here. We compared these two outliers here to the LG plus gamma 4 and found that that gave you a substantial difference. That means that a difference from the WAC to the LG here was quite substantial. If you went to LG plus I plus gamma, we again got a very substantial difference. LG plus I plus gamma 5, again a very substantial difference. LG gamma 14, again a big difference. And again here, a substantial, significant difference when we went from LG gamma 14 to LG R14. We also found that in the tree topology, that changed very radically. Even this small difference here led to differences in tree topology. Other details about the model of rate heterogeneity across sites was trimodal, meaning that it had three different, that model here with 14 rate categories had 3 peaks in it and troughs in between. It also was found that the topology of the optimal tree changed. The Robinson fold distance is 54, meaning that about, 27 edges in the original tree are not present. The tree inferred onto this model here are not present in the tree inferred onto this model here. So I mentioned early on that this is a homotacous model, or these were homotacous models. We've also developed models that are heterotacous. They were such models basically corresponds to scenarios like these ones here. I want you to consider a certain set of sites that may have evolved on this tree, other sets of sites that may have evolved on this tree. The topology is the same. And this case here, where the topology effectively is the same too, because this one here is embedded in the other two. In this case here, there may be different proportions of sites in these three in the alignment. And the heterotacous model essentially is then one that allow different sites to evolve using different tree topologies here that are similar to each other, yet somewhat different, and where those substitution models may either have equilibrium frequencies that are linked to each other or different. That is, that the sites in this group, in this group here may have the same nucleotide frequencies or amino acid frequencies, or they may be different. Or they may have different exchangeability rates that may be either linked or may be unlinked. That's a complex model, but it's at the same time, it's a model that's biochemically and evolutionary very, very realistic. It's one that biochemists really should nod in recognition at, because it brings back the reality of biochemistry and evolution into the phylogenetic analysis, something that have been missing for many years. It is, however, computationally a very parameter-rich set of models, and thus they require a substantial amount of time to execute. So have a look at an example here. In this case, we look at yeast data with these models here. I asked the question, how complex is the evolution model of evolution, or the evolution process, given what we think is a correct tree? In this case, we look at just 8 sequences of yeast. We look at second codon sites from a large number of sequences. And in this case, we use two programs called HAL and HAS from a paper that we published in Systematic Biology in 2014. In this case here, this is the tree topology that is assumed to be correct. And what you can see here is that we go from the root here, and here at the root here, we have 2 different edges here. And along these 2 edges, we have the same evolutionary process. Then that process change along this edge here to R3 and R2. Up here, R2 continues along that edge here and along that edge here. And at this point here, we change again to R1 to R1, and then it remains R1 over the whole tree. If we look at the proportion of variable sites, or proportion of sites, then we find that there's about 49% of the sites turn out to be invariable. The variable sites turn out to be divided into two groups. One we call V1 and another one we call V2. And the number of sites belonging to the first of these two is 39.6 sites and 11.3 sites. Now, if we look at the amount of change that occurred at those different sites, what we find here is that we end up having very different evolutionary scenarios here. For the first set of sites, the one that covers about 39.6% of the alignment, we see that evolution seemed to accelerate up along this edge here, up along this edge here too, and up along this edge here, and then it comes to a complete standstill along that edge, leading to these 5 species here. There's hardly any change occurring along those edges here. In other words, we see something that almost suggests that evolution first accelerates along that particular edge here, and then stops here while it keeps on going along this one here. On the other hand, for these other sites here, we see an acceleration along this, a much greater acceleration here than along there. An even greater acceleration along this one here, and continued evolution along these sites here. So in other words, we see a much more complex process operating, having governed the evolution of these particular sites in these particular set of genomes. This is something we have not really been aware of in many cases. We suspected that's the case, and it's possible now, especially not with HAL-HAS, but with IQ3, to do phylogenetic analysis of DNA or amino acids where these heterotachous models are being considered. It's a great advancement in phylogenetic analysis, and it's something I very much encourage you to push ahead with. Now, I think, oh yeah, this is kind of what next, and I think I'll stop at this case here. I'll just say that I'm working in these particular areas here where the red areas are, and I want to stop and say thank you for your time. I realize this has been a marathon effort, and you've been a fantastic audience. If you haven't died off, all of you, or if you're locked out, then it's absolutely fine, I understand. On the other hand, if you got something out of it, well done. Thank you.
Thank you very much, Lars, for the wonderful lecture. I have lots of questions, but maybe I'll mail you after that, because it's right on my field that I work using the applications, not constructing new models. But let's see if the audience has we also had some question. I'm also going to see the YouTube if we have questions there.
Good. Shall I stop sharing here and actually get people's faces back?
Yes.
Lots of compliments about the lecture.
Okay, I'm just going to stop.
João wants to ask something?
Want to keep? No, discard. Yeah. Yes, great, we're back. Wonderful.
Yeah. So I believe there is a way to handle these homotacos or heterotacos models using Bayesian inferencing. Yeah. So how can they compare it using program packages like these two or IQ3?
So when it comes to sequences that have evolved under heterotachous models, IQ3 is very much the software to go ahead with at the moment. That's not to say that there are not other methods that can do it. I suspect Nicolas Latillot in Lyon has some software that can deal with it as well. It's not unlikely that Mr. Bayes have implemented some features that allow you to do it. I'm not familiar with Bayesian phylogenetic methods to the same degree that I am with likelihood methods. And so I owe you a comprehensive answer. At the moment, I would suggest go ahead with IQ3. And if you found a result that you, or have several different results, and you want to test them a little bit more rigorously, especially if the DNA is data, data is DNA, then you can go ahead and use HAL-HAS, which is a little bit slower, but is perhaps a little bit more rigorous analytical approach that does not allow you to search tree space, but you can search model space far more carefully than IQ3 can do at the moment.
Thank you. So we can. So the survey that you talked about on the first version of the pipeline, on the first version of the approach, the new phylogenetic approach, is like adjusting our priors about the data set. Okay. And because we have some problems with priors. Yes, I did understand that.
Yes, I'd say the issue of priors is one that pertain uniquely to Bayesian analysis, and I do not know well enough how to choose priors in the Bayesian context. One of my former students, Simon Ho, who is now a professor at University of Sydney and has written widely on Bayesian methodology, is probably the right person to approach. Contact him and say that I have suggested you go to him and speak to him about how to select priors in your analyses when you're doing a Bayesian approach. I don't know how busy he is. I've lost contact with Simon, but I would say that he is the very best honors and master's student I've ever had in my lab. And he is the one that I recommended to the head of school when I left the University of Sydney back in 2009. He's a very, very approachable person.
Thank you very much. We have one question on the chat from R. Santarelli. Have you ever used time series comparison algorithms like ERP or DTW to assess how close a sequence is?
No, I haven't. I didn't even know that existed. I have to be very honest here.
Lars, I have a curiosity. How did you decide to call IQ3 IQ3? How did you choose the name?
I didn't choose the name. That was a name that was chosen, I presume, by Min Bui and Arndt von Hessler when Min Bui was a postdoc in Arndt's lab. Min is now a senior lecturer at the University, Australian National University, and is, getting himself established very well in an academic career there. He is also a very, very good person to talk to, a very, very knowledgeable person, and very helpful.
Okay. No curiosity about the choosing of the name. Just two letters.
I have had curiosity thoughts about that too, but I've never actually really had chance to. I should have. Well, I should have asked him, Min, because I have stayed with Min several times, both in Australia and in Austria, and I speak to Arndt von Hessler on a regular basis, and I could have asked, but it's always other things that are on my mind. I just need to send a text to my family saying that I'm just finishing here because they are a little bit worried that I'm not home yet.
Do you have any question?
Questions from the audience. If you'd like to ask a question, open your camera, your microphone, or write.
Well, it seems I've killed the excitement altogether.
Lars, I have a curiosity. When you are dealing with orthologs and you are doing trees of the genes, sometimes you have a gene of one species being positioned there. Let's pretend it's correctly alignment and etc. But sometimes the gene it does not reflect the phylogeny of the species. I saw that so many times. Like for bats, sometimes I had the gene alignment in the tree, like a sister of a mouse or a horse. And how do you interpret this, and are you worried about the comparative biology, and that sometimes a gene might be related to a species as a sister gene, and this does not reflect the speciation?
That's a really interesting question, and it's a very relevant question from a perspective of some work that I am doing at the moment here with some colleagues. So I was involved in bats, or the analysis of or publication of a bat genome paper in Nature in 2020. And I'm reanalyzing the data now with some of my collaborators there. What we have found is that among mammals, and there is a clear evidence that evolution has occurred heterotachously, and that means that for some genes we get one tree topology, for other genes we get another tree topology. If you read the Nature paper that I have referred to in the slides, if you read that, what you will find is that there is a clear evidence of inconsistency among the genes in terms of what the optimal phylogeny is. That was a big issue in our analysis, and we failed to come up with a clear understanding of why that is. So it's the very same question that you asked there. And I'm now asking, trying to solve that question, and I do that using other phylogenetic methods. And it looks as if it is due to heterotachy, namely that certain sites are evolving faster along some edges in the tree than other edges in the tree. And that gives rise to a very complex phylogenetic signal that most phylogenetic methods are not able to interpret correctly because they do not use models that are sufficiently flexible. That is,
could be.
So that's one thing. The other thing is that it could very well be that the evolutionary process along some of the different edges in your data has changed such that it's no longer, the data can no longer be assumed to have evolved under stationary conditions. You have to analyze them under more complex Markovian conditions. And that's the other thing that we know from the bat genome data paper, that a very large number of the genes had to be rejected because there was clear evidence that they evolved under heterogeneous conditions.
We did some observations on that, and maybe one point that could count is that sometimes a gene of two species, they decide to be more similar to the ancestor one.
Yes.
So then they will approach together. So that would be real biology, not an artifact.
No, it's absolutely.
Sometimes when they do that, they will position the species together in a tree, right? Even if they are in speciation, they are apart. And you know what we did? I was working with a colleague of João Paulo in Natal. And after some 3 months of working with orthologs and doing trees of all the proteomes, what we decided was to give up of tree. We decided to represent the phylogenetic, the similarity comparison as a graph. So if we took a bat, most of the genes of that bat has the closest orthologs of another bat, no problem. But when we analyzed the other bat says, "Oh, I'm sorry, but most of my genes have as the most similar in trees genes of horse." You know. So we just let it go. We draw as a graph. So we are being able of watching the graph and seeing what's the largest proportion of genes that are similar to what genus. So maybe for comparative genomics, of course, I respected the speciation. What you want to reproduce with the tree is the speciation, right? But for the comparative genomics, we thought that maybe graph could be a solution to represent the comparative biology. Yeah.
No, I agree with that. It's interesting what you say about the data. Sometimes we end up thinking that the data are wrong, and I guess from the take-home message from my talk is that it may actually not be the data that are wrong. It is our choice of methods that is wrong, and we tend to choose methods that we are familiar with, that we feel comfortable about using, and we are scientists, and so on one hand we are keen to explore, but on the other hand we are quite conservative in our choice of methods and are not prepared to explore other methods unless we're really forced to. And the worst of it all is that appropriate methods for many types of data may not actually exist yet. We need to develop those methods. So if there are anyone in the audience here that really is interested in method development, then I urge you to really get cracking, because there's an awful lot of new methods that needs to be developed. And sometimes we need to go back really to the drawing board, to the very beginning, and say, okay, fine. How can we do this better here? An example of that is something that I didn't present in the talk here, but it's something I'm just about to finish as a manuscript. And that is that there is a way of looking at multiple sequence alignments and figuring out which of those alignments are more appropriate for phylogenetic analysis. The problem that exists at the moment is that when we get a multiple sequence alignment and we look at it, some alignments will look more variable than other. But the reason why they look more variable is that we visually focus on sites that are conserved, that hasn't changed. And if that is the case, well, then we might actually fool ourselves big time, because our eyes gravitate to that column in your alignment where you see the same amino acid all the way through. That site may actually only be good in the sense that it gives us an idea that we got a correctly aligned site, so the alignment is correct in the neighborhood of that, it may be no good for us to assess how useful the alignment is in terms of phylogenetic inference. For that, we need to actually look at the variable sites in the alignment, and we don't have the mental or visual ability to comprehend that when we look at multiple sequence alignments. We are simply not trained to do that. And so we need software, and that's the software that I've just developed here over the years, and I'm now trying to get published and get out there. So and it could very well be if you survey your data with a new method like that, you might find that some of the alignments that give rise to surprising results in your data actually may have lost much of the signal, and that what you are depicting in a phylogenetic tree is essentially noise rather than a strong historical signal.
I wonder if it, I don't know, I don't have the solution, but I wonder if there is a way to filter the in such a way that you focus on speciation for some type of signals, which signals are doing good for telling us the speciation events. But for comparative genomics, that I think that there is a huge demand nowadays, because, for instance, in our data, if you took the gorilla, most of the gorilla orthologs, if you are going to study gorilla orthologs, you should pick human genes, not the chimp genes, because most of the gorilla genes are together in a tree closer to the human ones and not to the chimp ones. So for comparative genomics, people want to annotate and characterize a novel proteome. And sometimes the closest animal, the closest organism in tree, is not the one that's phylogenetically positioned there, right?
The problem with that, though, is that we need to go back to one of the early part of the slides that I gave you, is that if we use the tree as a guide as to whether the data and the analysis is good enough, then we run the risk that we get confirmation bias, because we let our prior expectation govern how we evaluate the data. That's not, I think, the best option. When we have scenarios where the different data sets give rise to different results, if the data are real and they are not fabricated, there are no errors in the true data, then it's really down to our skill and analytical tools to find out why do the two trees in a first pass, or first phylogenetic analysis, give rise to different trees.
I know. I have to repeat with different methods, maybe to bootstrap the methods to be sure. But I'm assuming that sometimes the similarity between genes should not reflect the speciation.
Well, in some cases, for example, with the hoatzin bird, that's a South American one you have in Brazil, isn't it? You know, if you look at some of its genes, you know, there's very striking evidence of convergent evolution amongst some of the genes. At least that was, you know, I haven't read about the hoatzin bird for many years, but, you know, when I did it many years ago, that was clear. You couldn't draw a phylogeny and you get what is assumed to be correct using several genes from, if the hoatzin bird was included in it. And, you know, obviously the hoatzin has developed a lifestyle where it gets its energy using certain enzymes that have co-evolved to a new way of actually supplying energy to the bird.
Oh, sure.
Yeah. anyhow, it's nearly 7:15 here, so I should really go home to my family.
Okay, Lars.
If there's a last-minute question, then drop it now. Alternatively, we can continue the dialogue by email if anyone have questions later on. And that's a possibility. If there are anyone who wants a copy of the slides as a PDF, then I can print it out as a PDF and then send it to you, Miguel.
Sure. I can post in the website.
Thank
you know
you.
that we didn't ask you, but can we provide the recording of your conference, because in the YouTube it's recorded.
You're absolutely welcome.
People would love to use it.
Yeah. No, you're most welcome. You're most welcome.
So, Lars, thanks a lot for coming, talking to us. It was very nice. The audience enjoys a lot. Many people are saying, yeah, great talk in the comments here in the YouTube too. Right.
Thank you so much. Very kind of you.
Yeah, thanks a lot. João Paulo.
Thank you very much, Lars. After I will have to ask my molecular phylogeny class to have new discussion to adjust to the approach that we have set. I learned a lot. Thank you.
We learned a lot.
I'm very, very pleased about that. I'm very pleased. It's a fascinating area.
Thanks a lot. Thanks you guys in the audience. We can move on, and thanks a lot, Lars. Thanks you guys. Bye-bye.
Okay.